Gram staining. What is gram staining? This staining technique was developed by Hans Christian Gram in 1884, so it is called as gram staining. This staining is a differential staining technique which differentiate bacteria into two groups. These groups are grams positive and grams negative. Principle of gram staining. Cell wall of grams positive bacteria contains high amount of peptidoglycan and low amount of lipids, so their cell wall is thick, where cell wall of grams negative bacteria contains high of lipids but low amount of peptidoglycan, so their cell wall is thin. Therefore, crystal violet iodine (CVI) complex formed inside the cell wall of grams positive cannot be decolorized by 95% ethyl alcohol or acetone alcohol, but the CVI complex formed inside the grams negative bacteria can be easily decolorized by them due to high amount of lipids in the cell wall of grams negative. So grams positive bacteria takes color of CVI and appears purple or blue, but grams negative bacteria takes color of safranin and appears pink or red. Procedure. Take a clean and grease-free glass slide. Make a thin smear at the center of glass slide by the help of inoculating loop from provided sample. Air dry the smear and heat fix. Add crystal violet to the smear and leave for about 45 seconds to 1 minute. Wash with distilled water or tap water. Add grams iodine to the smear and leave for about 45 seconds to 1 minute. Wash with distilled water or tap water. Add 95% ethyl alcohol or ethanol or acetone alcohol or 20% H2SO4 to the smear and leave for 1 to 2 seconds. Wash with distilled water or tap water. Add safranin to the smear and leave for about 30 seconds. Wash with distilled water or tap water. Air dry the smear and observe under the microscope. First under 40x, then 100x. Results. Grams positive, violet color, blue color, purple color. Grams negative, pink color, red color. Examples. Grams positive. Bacillus, Actinomyces, Corynebacterium, Streptococcus, Staphylococcus, Clostridium, Enterococcus, Mycobacterium tuberculosis, Nocardia. Grams negative. Escherichia coli, Shigella, Klebsiella pneumoniae, Fusobacterium nucleatum, Pseudomonas, Salmonella, Citrobacter.